Hello students, once again welcome back to my channel Biomania, and today we are going to study back cross, that is the second cross. As I have told you in previous video that there are three kinds of crosses. First one is the test cross, second one is the back cross, and the third one is the reciprocal cross. So today we will study back cross. Before studying the back cross, the first thing which comes in our mind, that is, why do we need to do the back cross? When we need to improve the breed, at that time we require the back cross. It means the one and only reason why do we need to do the back cross, that is for the improvement of the breed. When we need to improve the breed, at that time we do the back cross. Suppose that I'm giving you an example. Suppose that we are having two parents, that is one is capital T and lowercase t, and the next one is the lowercase t. So these are the two parent, and we want to improve that breed in that manner that in next generation we don't get any recessive phenotype, or you can say we don't get any short plant or animal, it can be. So what we will do for that, we will take any one from them and then we will cross it with the homozygous parent, and only homozygous parents which is dominant one. In the case of test cross, we are using the homozygous recessive because over there we are doing some kind of test, but here we are improving the breed. So we will take the dominant character, we will prefer the dominant trait. So here we are going to cross it with the homozygous dominant parent. So first we will see what kind of gametes. Suppose that we are taking this one. So what kind of gametes will form here? That is lowercase t and lowercase t. And in this case, there are these two gametes will form. Both are capital T and capital T. So what is the cross then? Okay. Here we are writing the recessive gametes, and here we will write the homozygous dominant parent gametes. So when it will cross with that, it will form like this. These progenies will form, and now here you can see there is all are heterozygous dominant, because in every progeny there is a dominant trait is present. So it means all progenies are tall, as their, you can say, as their phenotypic character, phenotypically they all are tall. We cannot see any short progeny here. So that is the reason why do we need to the back cross. When we need to improve the breed, at that time we need to do the back cross, and in that particular back cross we will make a cross with the homozygous dominant parent, and it can be used both in plants and animals. Okay, so that's not only the case of plants, it can be of plants and animals both. So I think now it is clear to you, and in the next video we will study about the reciprocal cross. Don't forget to watch that video because that is also very important. And thank you for watching my video. Don't forget to subscribe my channel and also share with your friends. So guys, thank you for this time. Bye bye.